Good afternoon. It's my pleasure to talk to you today about the natural history of Pleated disease, what the clinical needs are, and the needs for participation. Why is a natural history study important? The first and foremost reason why a natural history study of a rare disease is important is to help families and doctors predict how an individual child might do over time. One of the most common questions that parents and patients ask me when they come to see me is what to expect for the future. And both for what to expect and also for how to provide anticipatory guidance, I need to know how other people who have the same disease might have had developed complications over time or had development in their ability to do functional tasks like walking or use utensils or go to school, for example. Another important task that natural history study can fulfill is allowing physicians to understand what kind of complications might be seen in an individual rare disease. So just like your pediatrician, when you have a toddler, might warn you about the risk of exploratory behaviors under kitchen cabinets, or when you have a teenager, might warn you about excessive screen time. Anticipatory guidance for rare disease is also important to understand what organs might be involved or what kind of complications might ensue or when, and thus natural history study or an understanding of how the disease behaves over a larger group of patients can help us a lot in how to treat and manage an individual person. But as pertains to natural history, there are also significant implications of natural history studies for how to conduct clinical trials. The talks that came before this one really helped us have a view of what the future might hold for the therapy of Pleated disease. Well, there's no way to meaningfully judge how those therapies might work without a meaningful understanding of what to expect in an individual rare disease such as Pleated disease. Many of the grown-ups and kids in this room will remember Sesame Street and the recurring question of one of these things is not like the other. It's obvious to most that this little girl on the right up-hand corner is different because she's upside down. But there are other differences between these children. They're wearing different clothes, there are gender differences, and overall, it's important to know which difference it is that might be due to a treatment effect versus which differences might just be due to differences in the expression of the disease. These are all children, they look different, but what's important here is that the little girl is upside down. So the only way to understand what is the important difference to measure and monitor in a clinical trial, which is often called an outcome for a clinical trial, is to understand the natural history of the disease. More specifically, it's important to understand what a natural history study is. Natural history is often understood as a constellation of individual data points, and together each one of those individual data points form a clear picture of a specific disorder and how it might change over time, how it might be expressed differently in different individuals, and how the phenotype can be predicted by disease variables. Much like in the artist George Seurat's information on pointillism, where individual points of paint over time became a beautiful painting and a very detailed graphic image, natural history study data points where for each individual, there might be information about how they perform a specific motor task or what kind of complication they have. In a large group of aggregate data, might provide a very detailed picture of how the disease behaves over time. Importantly, we need to understand how a natural history study is performed. There are two main ways a natural history study might be performed. The first is a natural history may be performed by asking individuals to complete specific tasks, also known as outcome measures, in a standardized way over time, typically over many years, and observing this change over time. This is considered the gold standard of natural history studies. It's an important way to look in a detailed fashion at how a group of individuals can evolve over time in a very robust and reliable way, typically in a medical setting such as a hospital or an outpatient office. Unfortunately, these type of approaches may take years, and for a slowly progressing disorder like Pleated disease, even decades. Sometimes we can also perform natural history studies by looking backwards in time, using a time capsule of sorts by looking at the medical record. The medical record is collected in a detailed and rigorous way by physicians caring for the patients at the time when that medical record, such as a clinical note, is generated. It's therefore possible to rely on that medical record or a clinical note to generate a detailed view of how that patient was functioning or performing at that time, and for certain measures be able to then create a picture in a short amount of time about how a patient might have have evolved over a long period of time, such as since the disease onset, and a time period even spanning decades. Importantly, both of these approaches require patient participation in activities with no immediate benefit to them. One important thing to understand is that people might participate in a natural history study, and those same people might not be able to participate in a clinical trial. Sometimes patients can participate in both a natural history study and a clinical trial, depending on how those studies are orchestrated. It's also important to think through what might happen if a disease does not have a meaningful natural history study. Clinical trials cannot be started unless enough is understood about the natural history that outcomes can be selected. As a reminder, outcomes are those measures that allow you to understand what differences might happen because of the natural history of the disease and how the disease might behave without treatment versus how treatment might be affecting the expected natural history of the disease. Additionally, if there aren't detailed enough natural history studies, trials might reasonably need to enroll more patients. They won't be able to predict well enough what might make meaningful differences and how to measure them, and they might need to plan for more patients than what would be needed if a detailed natural history study was available. It's also important to note that out of 250 publications in the last 10 years mentioning Pelizaeus-Merzbacher disease in some fashion or PLP1 gene mutations, I found only 7 that have any data on cohorts of patients with Pelizaeus-Merzbacher disease that mention more than just 1 or 2 individual patients, and none of these truly include a natural history study. So unfortunately, we have quite a dearth of information in Pelizaeus-Merzbacher disease overall. So importantly, what are we doing within the scientific and advocacy community to support PMD? The first is that we're conducting a retrospective natural history study that's funded by the PMD Foundation. This approach is the shortcut approach I described earlier. It's based on something called annotated medical record collection and extraction. We take medical records, and in a detailed fashion, like you might annotate a favorite book, we make notes to document when specific changes happened that are important to the disease, such as learning certain skills or losing certain skills. This approach allows us to extract standardized data from the medical record and allows us to explore expectations around the development of key disease features. It will help us develop a framework to understand how key outcome measures might be able to be applied in Pelizaeus-Merzbacher disease. I want to give you some examples of how retrospective data collection has already begun informing our ability to understand leukodystrophies. On the right, there's information about HABC, which is another hypomodeling leukodystrophy. In that study, we have over 150 individuals, and we're in the process of doing a detailed analysis of developmental milestone acquisition. Importantly, because we have a lot of patients, we can provide very granular stepwise information about how individual groups might behave. So in this study, we can generate whether or not people might be more likely to acquire specific milestones if they present early or late, and what might happen if they have specific gene changes. We're starting to be able to see that information also in Pelizaeus-Merzbacher disease in the images on the left. But we only have 34 individuals, and so those stepwise differences are still very big. And it's hard to predict entirely how the population might be behaving when in each cohort you only have 1 or 2 patients. For example, there's only 1 PLP null patient in this cohort, and the other mutation groups also have limited amounts of patients. So overall, it's important that we have more patients able to participate in these studies to allow us to get more granular and detailed and reliable data. I also want to show you other types of data that we can acquire from retrospective data collection. This again is the beginning of our data on PMD, and you can start to see that we're able to understand how, depending on at what age people present, how they might have different disease features. So you can see that the younger patients in the graph on the left are more likely to accumulate a lot of different types of symptoms versus the patients who present older after 2 years of age might have much milder disease symptoms overall and might only, for example, have some problems with spastic diplegia or cognitive symptoms. Similarly, we can map out, according to genotype, how long people might maintain certain levels of motor function. So we're applying a new scale that we've called the GMFC PMD, and we're able to demonstrate that patients with deletions so far look like they're maintaining ambulation and certain motor skills for a longer amount of time. Right now, this is done based on approximately 65 patients collected in the study. We're expecting larger amounts of enrollment from additional centers in Canada, Japan, and the Netherlands in the coming months, and additional participation will be necessary to get a detailed view of how these diseases perform. This is another example of the amount of detailed information that one can get if you have access to a greater number of patients. These are examples of the same type of graphs that I just showed you for Prader disease in metachromatic leukodystrophy. In this study, we have over 150 individuals, and you can see that we're starting to get detailed information about how people develop over time as far as their ability to maintain ambulation, depending on whether or not they presented at a certain age, that's the graph on the left, and whether or not they presented primarily with motor features or with just cognition, or with both motor and cognitive features. So overall, much like the pointillism pictures, where the greater the number of points, the better and more beautiful the data and picture that evolves, this is true also for data collection in natural history studies, where the greater amount of data and the greater amount of patients who participate provide us with a much more detailed view of how the disease might progress. So what are we doing to support Pelizaeus-Merzbacher disease and the need for Pelizaeus-Merzbacher disease natural history? The second thing that we're doing is looking at a prospective natural history, like the gold standard natural history study that I described earlier. This involves detailed in-person outcome assessments or motor and cognitive tests done on individual patients over time, typically in an in-person setting. This allows proof of whether certain tools are able to be measured in certain disease populations, and helps validate the use of these tools for future clinical trials. This type of work is typically necessary in order to be able to use tools and outcomes for clinical trials. Obviously, this looks different during the time of COVID-19, and there are modifications in place to currently planned natural history studies to permit for remote assessments in the context of the need for social isolation. We are doing this right now as part of the Global Leukodystrophy Initiative or Clinical Trials Network, or GLIA-CTN. This is an NIH-funded consortia with sites distributed across the U.S. Each site will be able to assess individuals with PMD according to standardized protocols. And specifically, we have projects around this longitudinal natural history study across leukodystrophies, but specifically in PMD as part of this project. So what can you do to support PMD? The main thing you can do right now is participate in a natural history study. Find the right fit for you. There's no contribution too small to make a difference, just like there's no dot on a beautiful pointillism painting that doesn't contribute to the overall picture that is painted. You can share medical records, imaging, or genotypes. You can travel to a participating site for in-person evaluations, or during COVID-19, participate in remote assessments. Or you can contribute to research by sharing your impression of how the disease impacts you as a person and as a family through surveys. How can you do this? One way is to contact your disease foundation, in this case, the Pelizaeus-Merzbacher Disease Foundation. You can also contact GLIA-CTN or a local international partner of the consortia by going to www.theglia.org. Go ahead and look for the sign that says collaborate, and then ask to participate. There'll be detailed information for how you can contribute to these efforts. It's important to know that together we can be ready for the promise of clinical trials. One piece of data at a time, we can support PMD through its need for developing therapeutics. Thank you very much, and I'm happy to take any questions in the later session.